right, let's see here. We want this one for tonight. All right. So like I was saying, I want to go back and hit on a couple of the DNA replication points that are going to be important for the future. So again, DNA replication is all about copying our DNA. So all the cells in your body have the same set of DNA in it. So each time that we split and copy a cell, we have to make a copy of that cell's DNA, which is what replication is all about. So DNA, we already talked about at the beginning of last week. It's this kind of helix structure, this twisty structure with the phosphates and sugar groups going along the outside. And the little pieces of the ladder here on the inside are our bases. So those bases are kind of the main things that we are actually copying when we are replicating our DNA. So one of the first things we talked about with DNA replication was that this idea of semi-conservative, right? So each of the daughter DNA strains, each of the new DNA strands that we make is made up of one copy of the old strand and one copy of the new strand. So the old strand kind of acts as a template that the new strand then gets copied on top of. So the old strand is where we have our C's and our T's and our A's, and then we copy and match up those with their G's and their A's and their T's on the new strand. So that's what semi-conservative means. It just means that the new pieces of DNA have one old and one new strand in each. That stuff wasn't super important. So the first major thing that we talked about with our DNA enzymes, so there's 4 different enzymes we talked about: helicase, primase, polymerase 3, and polymerase 1. So helicase is kind of the simplest of all these. It's responsible for unwinding and splitting up the DNA strand. So before, that DNA was all twisted together and all the bases were connected with each other. And then helicase comes in and splits the strand apart. So it's sort of like the DNA zipper. It unzips and opens up our DNA. So that's enzyme number 1, helicase. The next enzyme was primase, and this guy is responsible for putting down those RNA primers. So this was that question that I posted in Google Classroom yesterday, was about why we need these primers. And the reason is that we have to give our new DNA a starting point to start building off of. Right? We have to have sort of like a base to start building. Our RNA primer is used as that starting point. So we can't start copying until we get this RNA primer put down. And primase is the enzyme that does that. So still relatively straightforward, right? Primase puts down the primers. So our little starting points here that are made of RNA are where we can start making the new DNA. And again, semi-conservative because we have the old strand, the dark blue in this picture, and then the new strand, the light blue, is what's going on top. So our final DNA has the one original strand and one new strand. So helicase opens up and unwinds the DNA. Primase puts down these little primer starting points. So when we start making DNA, we have to make DNA in what's called the 5' to 3' direction. So there's different sides of the DNA that we're not really going to worry about exactly why they're called 5 or 3'. But there are these different sides that make the DNA go in one direction or another. And so when we're building DNA, it has to be in this 5' to 3' direction. So when we're building our DNA and we have the helicase that's opening up the DNA strands, what we end up with is one side of the DNA that can just be built continuously in one long string, one long direction from the 5' end towards the 3' end, which is constantly being opened by helicase. This is called the leading strand. So the leading strand of DNA is this one where it just needs the one little primer to start off with, and then we can start building from 5' to 3', no problem, very easy. Our other strand, our lagging strand of DNA, we don't have it so easy over here. We end up with lots of these little short segments called Okazaki fragments, because in the lagging strand of DNA, the DNA keeps trying to be built in the opposite direction than the DNA is being opened. So the new DNA is being built to the left, but the old DNA it's based on is being opened to the right in this case. So we have to put down a primer and then fill in some DNA and then jump back to the newly opened DNA, put down a primer, fill in some DNA, and then jump back, put down a primer, fill in some DNA, and so on and so forth. So that's what makes this the lagging strand, is we're constantly putting down all of these little short fragments. So leading strand, smooth, easy, one long, big piece. Lagging strand, short fragments being built in the opposite direction from where our helicase is going. So who's dealing with all this DNA and RNA that's being put down on the two strands? Those would be our two DNA polymerases. So these are those final two enzymes that I had you guys write about. So polymerase 3 is actually sort of like the main dude, the main guy to copy DNA. So polymerase 3 is putting all of that leading strand DNA down. So all of this new red DNA down here, polymerase 3 is putting all that down. And it's also responsible for filling in these gaps up here on the lagging strand. So the new DNA that initially gets put down on the lagging strand, that is polymerase 3 as well. So this guy does the vast majority of the DNA copy, the actual replication part. Polymerase 1 is only responsible for taking away those primers and filling it in with DNA. So all these little red primers here, those are made of RNA, which is a different type of nucleic acid, which we'll actually be talking about a little bit later tonight. We don't want that in our DNA, right? Only DNA should be in our DNA strand. No RNA allowed. So we got to get rid of this RNA primer. That's where polymerase 1 comes in, basically takes out the RNA and puts down a little bit of DNA to replace it. So polymerase 3 is doing all of this leading strand filling in, and most of filling in between the fragments of the lagging strand. And then the only thing that polymerase 1 does is come in and take off the primers and replace it with DNA. So it just swaps out the RNA for the same type of DNA. All right, and then again, our replication fork, so that DNA split. In this picture, it's going to the left. So our top strand of DNA, in this case, is the leading strand because we're constantly just making a smooth, even flow of DNA all the way down the strand. We've got our 5' end over here, and we're building towards the replication fork, towards the 3' end over here. So that's our leading strand. Our lagging strand is unfortunately flipped around. So our 5' ends are over near the opening. And so we build all these little fragments out towards the 3' end, and then jump back and fill in the fragments and jump back and fill in the fragments as this replication fork opens up new DNA that needs to be copied. All right, so the main things to know about DNA replication is those 4 different enzymes and what they do. So the helicase, primase, polymerase 1, and polymerase 3, as well as the difference between the leading strand and the lagging strand and why those are different. All right. Any questions about the DNA replication? Anything that you want me to go back and talk about a little bit more?
Yes, sir.
All right, thanks, Fernando. Anybody else, any questions about any of that? Anything that you want me to go back and hit on, make sure that you got?
No.
All right, thanks, Paula. All righty then. In that case, we shall move along to our, there we go, main topic for tonight. Which is the Tuesday notes for today on protein synthesis. So synthesis is just a fancy science word for building or making things. And so protein synthesis is all about how we make different proteins using the code in our DNA. So we're taking our DNA code, turning that into RNA code, and then using that to make proteins. All right. All right, so a little refresher on DNA, or a little review of what we can use DNA for. So all of those bases that make up your DNA, so the A's, the C's, the T's, the G's, certain segments of your DNA, so certain sequences of those bases, are known as genes. And your genes are basically what control the running and organization of your body. So the genes, your genes are super duper important. The way that they run and control your body is because these genes are different codes for what are called polypeptides, also more commonly referred to as proteins. So all the different proteins in your body, including the enzymes that we just talked about, so helicase, the polymerases, and primase, those are all different types of proteins. And the code to make those proteins lies in the genes that make up your DNA. So proteins do all sorts of different jobs in your cells. They're used to build new cells. They're used to tear down pieces of old cells. They're used to create energy for your cells. They're used to help copy your DNA, like we were just talking about. They're used in your stomach and your digestive system to help break down food and convert it into energy. Proteins are like the workhorses of your cells. They do tons and tons and tons of different important jobs for the cells in your bodies. And all the proteins, the code to make those proteins is in the genes of your DNA. So these genes are these sets of basics. So like ACCGTTGTACCTGCA, that could be a gene. And that gene is the code for one specific type of protein. So today we're going to talk about the first step of how we take DNA and turn it into RNA, which is sort of like the message about how to make the protein. And then on Thursday, we'll go over the process for how we're able to take that RNA message and actually convert it into a protein. So proteins are made out of smaller structures called amino acids. There's 20 different amino acids that exist on planet Earth. Luckily for you guys, you don't have to remember really much of anything about the amino acids, unless you go to college and take a microbiology course like Mr. Stanley had to do, in which case you get to memorize the names and structures of all 20 of these amino acids. So at some point in my life, you could have told me to draw phenylalanine, and I'd be able to draw out this structure. Or you could have shown me this structure right here and asked, what amino acid is this? And I'd be able to tell you, oh, that's a serine. So you guys luckily don't need to do that, because that is just way too much work that nobody needs to do. But we make proteins by stringing together a whole bunch of amino acids. Either dozens or possibly hundreds of amino acids go into making one protein. So they're kind of like the building blocks of our proteins. So each amino acid has basically this same structure. We have this kind of central carbon atom right here. Then on every amino acid, one side of it is this COOH group. The other side is this NHH group. The third side is just a single hydrogen. And then the fourth side is some sort of side chain. So this side chain right here is what is different between each amino acid. So this could be like some sort of ring or some sort of long carbon-based structure, or there could be some sulfides thrown in there. So this is different depending on the amino acid. But these kind of three structures, this amino group, carboxylic acid group, and hydrogen, are the same across all of them. So when we combine dozens or hundreds of these amino acids together, we are able to end up with a protein. So we basically turn these amino acids into these big long chains, which get folded up into a protein. So we start off with the amino acids kind of floating around by themselves. Then, based on the gene that we have, we take those amino acids and put them in a certain order in this big string. And then we fold up that string in a certain way, and that is what gets turned into the protein. So a gene essentially just tells us what is the correct amino acid order that we need for this protein. So for example, whatever this gene was that we're turning into a protein, it said, all right, first you need a blue amino acid, and then a brown one, and then a green, and then yellow, and then purple, and then green, and then blue, and then black. That's the order of amino acids that we need. And then once we put all those amino acids together in this polypeptide chain, we took that chain, folded it all up, and made our protein. So obviously DNA is where our genes live. And inside of the cell, DNA is found in a special compartment called the nucleus. So the nucleus is sort of this big central structure inside of our cells, and it helps to protect the DNA. So obviously your genes are extremely important to keep them intact and keep them protected. So the DNA isn't just floating around kind of willy-nilly in your cell. It's packaged up inside the nucleus, and then the nucleus is what is inside your cell. So your genes and your DNA live inside of the nucleus. There is one small but significant problem with that, though. So we've been saying how we're going to use our genes in order to make the proteins that our body needs. Unfortunately, proteins are made by structures in your cell called ribosomes. So we'll definitely be talking about these guys later on. The problem with that is that the ribosomes are not inside the nucleus. The ribosomes are all kind of floating around in the cytoplasm, which is just sort of like the filler inside part of the cell. So the ribosomes are inside the cell, but they are outside of the nucleus. So they might be attached to some structures. They might be kind of free floating outside. But the main issue is that the DNA and the genes are inside the nucleus. The ribosomes, where we make the proteins, are outside the nucleus. So we have this barrier of the nucleus in between the DNA and the ribosomes. So we need to turn these genes in our DNA into proteins, but the genes are inside the nucleus and the protein factories, the ribosomes, are outside. This is a little bit tricky. The DNA can't leave the nucleus in order to get the genes read because like we were saying, like I mentioned earlier, the DNA is too important. It could be damaged if it leaves the nucleus. So the DNA has to stay inside the nucleus. So instead of taking the DNA out of the nucleus, we make a copy of the gene. So we copy the DNA code, and then we take that copy out to the ribosome so that it can be turned into a protein. That way, we can keep the DNA inside the nucleus and keep the ribosomes outside the nucleus and just take this copy in between the two. So we take the copy from the DNA, it leaves the nucleus and goes out to the cytoplasm where it can be copied by, or read, by the ribosomes. And then the ribosomes are what will make the proteins. So like I said, synthesis is a fancy science word for building or making stuff. And so since we're using ribosomes to make proteins, we call this process protein synthesis. So copying the DNA's code inside the nucleus, carrying it outside the nucleus, and have it be read and turned into a protein by the ribosome, the whole process is called protein synthesis. So tonight, we're going to focus on this first part right here of copying our DNA code. Anybody have any questions about genes, DNA, amino acids, ribosomes? Any questions on anything that we've talked about so far?
No, mister.
No, mister. No.
All right. Cool. Thanks, guys. Appreciate it.
Mm-hmm.
So, how do we make a copy of our DNA? That is where DNA's smaller cousin RNA comes into play. So DNA is the master plan. It's the original. It has the original masters. RNA is the copy. It is the blueprint of the master plan. So if DNA is our original set, RNA is how we copy that gene so that we can safely bring it to the ribosome, which is outside of the nucleus. So I talked about this a little bit last week, but DNA and RNA are slightly different from each other. And there are three key areas where they are different. They have a different sugar on them. They have a different strand setup, and they have slightly different bases on the inside. So we'll talk about each of those one by one. So DNA, if you remember, is a big fancy word that stands for deoxyribonucleic acid, whereas RNA is just regular ribonucleic acid. So the first major difference between RNA and DNA is that RNA has, for its pentose sugar, because remember, each nucleotide in DNA and RNA needs a phosphate, a sugar, and a base, RNA has a slightly different sugar from DNA. So the RNA sugar is called ribose, and that's where the R comes from in RNA. The DNA sugar is called deoxyribose, and that's where the D comes from in DNA. So you don't have to know exactly why they're different. I'll show you really quick. So here are our two sugar molecules, our RNA ribose and our DNA deoxyribose. And you can see on our number 2 carbon over here, ribose has an OH group on that carbon, whereas deoxyribose only has an H. So the DNA sugar is missing an oxygen. It is deoxygenated, which is where the deoxy part of deoxyribose comes in. You guys don't necessarily need to know that. You just need to know that the sugars are different. The RNA sugar is called ribose, and the DNA sugar is deoxyribose. So that is difference number 1 of 3. Difference number 2 of 3 is that they have slightly different bases. So we talked about the DNA bases, A, C, G, T. And remember, actually, no, I want you guys to tell me. So how do the bases in DNA pair up with each other? Which bases go with which? Does anybody remember out of A, C, G, and T, which bases always go together?
I know A goes with T, C goes with G.
Yep.
So it'd be C, G, C, A. On the top, the bottom on.
Oh, yeah, right. So yes, Pete is 100% correct. So T's and A's and T's will always go together. And then C and G and C will also always go together. So AT or CG, those are the different pairs that we have. RNA is a little bit different because RNA does not have T. Instead, RNA has U. So when we're making RNA, instead of matching a T with an A, we would match that
A and U.
A U.
Okay.
So instead of being ACGT, RNA is ACGU. No T's in RNA. If you see a T somewhere, then it has to be DNA. It can't be RNA. RNA has U. So that's difference number 2. T's in DNA, U's in RNA. Finally, difference number 3 out of 3 is that DNA is double-stranded. So we talked about how it's the 2 strands that kind of wrap around each other and twist around in that helix spiral. RNA, however, is single-stranded. So it's only one long strand for the RNA. It doesn't have the double strands that match up in the middle. So yeah, so here's a good illustration. Over here, we've got our DNA. It's got that deoxyribose sugar. We have the 2 strands wrapped around each other. And we have G's, A's, C's, and T's in DNA. Whereas in RNA, we have G's, A's, C's, and U. So no more T's, we have U, uracil. And RNAs are just single-stranded. So the one single strand with that ribose sugar or the backbone, that is what RNA looks like. So between DNA and RNA, they're similar, but they have different sugars. They have a different set of bases, with DNA having T and RNA having U instead. And they have a different setup for their strands. DNA's got the double strand, RNA's got the single strand. All right. So, like I was saying at kind of the beginning of the notes, we have our DNA that's sitting nice and cozy and safe inside of the nucleus. It's got all its genes going up and down the DNA. But if we want to turn those genes into proteins, we've got to take that information out of the nucleus and over to the ribosome, where the proteins get made. We can't send the DNA out of the nucleus because that would be dangerous. Instead, we have to make a copy of this DNA using the RNA. So yep, just a reminder, DNAs has T and A matching up and C and G matching up. RNA, C and G, still the same, not different, still also matching up. And now, instead of matching with a T, our A will match with a U. So there are 3 different types of RNA. Messenger RNA, also known as mRNA, is kind of the most important one and the one that we'll be focusing on the most, especially today. tRNA are also somewhat important, and we'll talk about them a little bit more on Thursday. They're basically what are responsible for sort of shuttling those building block amino acids back and forth. And then finally, ribosomal RNA is a special type of RNA that makes the ribosomes. We're not really going to focus on that one today or Thursday, so don't worry about that one so much. But mRNA, that's what we're really going to be working with today. So the mRNA, the messenger RNA, is called that because this is what carries that DNA message. So that thing that is leaving from the nucleus and going into the ribosome to be copied, that is the messenger RNA. It copies the code from the DNA and carries that code over to the ribosomes. So when we're getting ready to make a specific protein, we'll use the DNA to make a strand of messenger RNA. So the gene of a DNA will be copied onto a strand of mRNA. Then when the mRNA goes out of the nucleus to the ribosome, that copy will be the code for a specific protein. So we'll talk a lot more about codons in our lesson on Thursday, but essentially every 3 bases, every 3 letters of the mRNA is called a codon, and it's called that because it is the code for a specific amino acid. So we'll talk about more how this works on Thursday, But the basics of it are every 3 letters tells the ribosome, now this is the protein that I need, or the amino acid that I need. Now I need this one. Now I need this one. Now I need this one. There are some that aren't specifically used for amino acids, like these UAA, UGA, UAG are what's known as stop codons. So they are what tell the ribosome, stop, we're done, no more amino acids needed. There's a lot of proteins that start with AUG, so sometimes people call that the start codon. But again, we'll talk a lot more about codons and how they work on Thursday. So we copy the DNA onto the mRNA, and then the mRNA kind of splits off from the DNA and leaves through little holes in the nucleus called nuclear pores. Again, it has all the same bases except where you would have put a T for the DNA, you put a U for the mRNA. So it's kind of hard to picture this with just me talking about it and using words. So here is kind of a picture of what this looks like. So it's sort of similar to DNA replication, where we're opening DNA and then copying the different bases. Only in this case, once we're done copying the DNA, we don't end up with 2 new sets of DNA. We end up with this little string of mRNA, and then the DNA closes back down. So basically, it opens up, gets copied, closes down, and then the mRNA heads out. So we make a copy of our DNA, just like we would if we were going through DNA replication, where we just match the DNA kind of template strand with RNA bases. So just like you guys were doing on Tuesday of last week when you were making the new DNA strand from the base DNA strand, a really similar thing is going on right here. So we've already kind of started one in progress right here, but if you can see where they're matching up, on the DNA template strand, we have a T, and so that means that for our mRNA, we're going to have an A. On our DNA strand, we have an A, so we match up not with T, because remember, no T's in RNA. Instead, this DNA A gets matched with an RNA U. If the DNA has a C, the RNA will have a G. If the DNA has a G, the RNA will have a C. Again, if the DNA has an A, the RNA will have not a T, but a U. G on the DNA gets matched with C on the mRNA. A's on DNA get matched with U's on mRNA. And then T's on DNA get matched with A's on mRNA. So originally, for the first part of this mRNA, the DNA would have read CGTTGG, which makes our mRNA GCAACC. So again, the DNA opens up this RNA polymerase. So now we don't have DNA polymerases anymore because we're not making DNA, we're making mRNA. So the RNA polymerase goes through and builds an mRNA strand using the template of this opened DNA strand. So that's sort of how we go through this initial process of converting the genes in our DNA into a form that can be taken out and leave from the nucleus. So this whole process that we're going through right here is called transcription. So we're transcribing DNA into mRNA. So we're converting it into a slightly different form so that this mRNA can leave out of the nucleus and go to the ribosome. So I actually think I have skip through some of this stuff. I have a video, I think it's this one. So my apologies, at the beginning, it's going to be a little bit kind of jerky looking for you guys, but I think it does a good job of showing sort of the basics of this initial part. So here we have our big cell, and we have all these different little pieces, but the nucleus is hanging out right at the middle. And again, the nucleus is where our DNA is. So it's all bundled up into things called chromosomes, which we'll talk about way later on in the term. And it's wrapped around these things called histones. Don't worry about that. But here is our DNA strand, right? We got our double-stranded DNA, and certain sequences of this DNA are a gene. So it could be a couple dozen bases, it could be a couple hundred bases, but this one gene contains the code in it for one protein. So we get this big old guy called RNA polymerase that opens up the DNA and starts making mRNA. Again, we're making our mRNA just by matching up with our DNA bases. So wherever the DNA has a T, the RNA is going to have a G. Wherever the DNA has a G, the RNA will have a C. If there's a C on the DNA side, there'll be a G on the RNA side. And then the only major difference is that if our DNA has an A, normally A's get matched with T's, but in RNA, we don't allow that, no T's in RNA. It gets matched with a U instead. So we're building this big long strand of the mRNA. And then as we're building it, after the bases get matched up and paired together, you can see at the end, the strand will start to kind of peel off as it's getting made. So we have our strand kind of peeling off the DNA as we move along and make an RNA copy of all of our different little bases down here. So now we have our messenger RNA, our mRNA, this big long strand with the ribose sugar, single-stranded with U's. Don't worry about the stuff that it's doing right now. After the mRNA gets made, it gets like chopped up in a certain way. We're not going to worry about that. But once the mRNA is all ready it can leave from the nucleus. So remember, this big purple lumpy thing right here is our nucleus. Our DNA has to stay inside the nucleus. It can't come out, otherwise it might be damaged and that gene might get damaged. And that's very bad. We don't want damage to our genes. So that's why we get this little mRNA strand to come in, make a copy of that DNA, match up all the T's with the A's with the U's, the C's with the G's, and the G's with the C's. And once we make that copy of the DNA strand with our little mRNA single strand, then it can leave from the nucleus and head on over to the ribosome, which is our little protein factory that is going to be building a new protein based on this mRNA code that is coming in that got copied from our DNA. That is the process of what is called transcription. Copying a gene in the DNA onto a strand of mRNA that can then leave from the nucleus and go make a protein. All right. Anybody have any questions about DNA versus RNA, or how we use the DNA in order to make this mRNA strand? Any questions, any concerns, anything that you want me to go back and talk about a little bit more?
No, mister.
No.
All righty. So that is where we will kind of pause today for the notes. Like I said, that's sort of the first half of this process. And then on Thursday, we'll get into the second half where we'll talk about how, once this RNA makes it to the ribosome, how we actually turn this big long string of bases into a brand new protein. So we'll get into that on Thursday. But for the assignments for today and tomorrow, again, since you guys are here in person on the Zoom call, you already have your attendance credit for tonight. And so if you want to work on the Tuesday assignment sometime later, that is fine. But essentially, all you have to do for the Tuesday assignment is write up what are the three major differences between DNA and RNA. And so what are the things that you can use to distinguish between what is DNA and what is RNA? There are three of them that we talked about tonight. And so just listing those and describing a little bit about what makes them different in these three different ways is the Tuesday assignment. And then the Wednesday assignment that is going to be posted up on Wednesday, obviously, looks very similar to an assignment that you did last week, where we have a string of DNA bases. Only this time, I don't want you to turn it into a complementary DNA strand. I want you to turn it into the complementary mRNA strand. So if this was the strand of DNA that is being copied, then what would its matching RNA strand be? So remember, you're matching your C's with your G's. You're matching your T's on the DNA to A's on the RNA. And then the only major difference is that normally where you would put down a T on the DNA, for RNA, you put down a U instead of the T. But other than that, it's extremely similar to the earlier homework that you had, where you had to copy a new DNA strand, only this time you're copying a new RNA strand. So that is the assignment that will be posted up for your attendance credit for tomorrow. Where am I at? All right, guys. So does anybody have any final questions about DNA or the transcribing it into the mRNA, or the differences between the DNA and the RNA, or about the different homework assignments for today?
No. No, mister.
No.
All
No,
righty, guys. Well, if you ever do have any questions,